I am interested in looking at sex-specific differences, specifically in neurodegenerative disorders. And so here I'm showing 3 different neuropathological stagings of 3 different neurodegenerative disorders. And you can see they're quite different in terms of where the origin of the pathology starts and where it spreads to. But what's common amongst these 3 diseases, so Parkinson's disease, dementia with Lewy bodies, and multiple system atrophy, is they're all grouped under what are known as synucleinopathies. And the grouping of synucleinopathies just simply means that the primary pathology is the intracytoplasmic accumulation of proteins, such that the primary component of these proteinous aggregates is the alpha-synuclein protein. And so there's been a lot of focus on what alpha-synuclein is, and we know it's small, it's highly soluble, it's natively unfolded, but its precise function is still not quite known. But nonetheless, it's present in many neurodegenerative disorders. And what's thought to be happening is that there's this prion-like spreading hypothesis, so such that normal alpha-synuclein somehow gets misfolded, starts to aggregate, forms the toxic fibrils, and it's believed that it can propagate from one neuron to another. And so as Millar said, this has been extensively studied. There have been postmortem studies, as well as cellular and animal models in which have investigated the spreading. But specifically, there's been most recent work examining the spreading using neuroimaging. And so here I'm highlighting a specific group, Dr. Alan Dagger's group at the MNI, in which had shown that there's different degrees of pathology in patients with Parkinson's disease. And what they saw was that there's a high spatial overlap between these atrophy patterns and resting state connectivity patterns, as well as intrinsic connectivity networks. So essentially, seemingly that this prion-like spreading hypothesis is really spreading via the brain's connectome. And so we wanted to examine this. And of course, we're using animal models to examine alpha-synuclein propagation because we can really time and examine progression over time within these mice. So Millar mentioned this M83 transgenic mice. So these are mice that have the human alpha-synuclein transgene, but they also have the normal endogenous mouse alpha-synuclein. But essentially with the human alpha-synuclein transgene, we also introduced the A53P mutation. So this is a point mutation found in one of the familial forms of Parkinson's disease. And essentially with this mutation, it increases the propensity of the protein to form intraprotein interactions, therefore causing it to misfold. And as I said, once we have misfolded alpha-synuclein, it's believed to aggregate and then form the toxic fibrils. So now with this mouse model, what we can do is we can actually inject the mice with the fibrils, and this allows us to have a known disease epicenter from where we can examine the spreading. And so the experimental design that we used is, again, using the M83 mice. They have only one copy of the transgene, so they're homozygous, and of course, using both male and female mice. And so, as I mentioned, we inject the mice with either saline, PBS, or the human preformed fibrils of alpha-synuclein in the right striatum. And we wanted to examine the propagation over time, and so we required MRI at multiple time points. So specifically, an in vivo T1 weighted scan at 100 micron voxel resolution. So we acquired the MRI at 1 week prior to the injection, and then at 30 days post-injection, once alpha-synuclein pathology is present in terms of postmortem staining, at 90 days post-injection, so once motor symptom onset is nearing, and then at 120-day post-injection, so once the mice are fully symptomatic and nearing their endpoint. And so here I'm just showing the number of mice that we acquired per group, per sex at each of these time points. As you can see, we siphoned off mice to perform the postmortem analyses. And so at the end, we have about 8 mice per group to investigate the changes over time. And so we use deformation-based morphometry, which essentially allows us to examine differences at the voxel-wise level. And as Millar mentioned, we adapted this technique to have a longitudinal way to examine the trajectories of change over time, where we first average all of the subject time point scans together to create subject averages, and then we average all the subjects to create a population average. And this now allows us to have a common space from where we can perform our statistics, such that no matter what voxel I point in the population average, it's that same voxel in every subject in every time point. And so what we did first was we performed linear mixed effects models at every voxel in the brain using the deformation fields. And so here I'm showing the results for when we examine the PFF-induced areas where we see differences in trajectories over time in terms of volume. So here I'm showing the effects for the M83 mice that receive the fibrils compared to M83 mice that got the saline injection over time. So looking at this two-way interaction. And so here's coronal slices from posterior to anterior slices, and the color maps are denoting significant changes in terms of the trajectories between these two groups. And so we take a look at this map, we see our injection site where we see this difference in terms of the trajectories, but also in regions that have known projections to and from the right striatum, such as the frontal cortices, as well as the basal ganglia system with the caudate putamen, the globus pallidus, as well as some thalamic areas, and then posterior midbrain areas, and as well as brainstem areas. And so if we take a look specifically at a peak voxel of interest in our injection site, here I'm showing the trajectories over time, so the relative volume across the number of days post-injection, where the blue solid line is the saline injected mice, and the dashed red line is the M83 mice that received the fibrils. And so as you can see by the red dashed lines, we see the steepest rate of decline in the mice that received the fibrils. And this nicely went along with when we examined the survival of our mice. So here I'm showing percent survival across the number of days post-injection, such that you can see only the mice that received the fibrils succumb to their disease within the time frame that we were looking at. But when we took a deeper look at who these mice were in terms of male and female ratio, we actually saw that it was only the male mice that succumbed to their disease. So that's shown by the light blue color and for the males and the pink color for the females. So this was striking, but not striking, because we know that there's higher prevalence of synucleinopathies in men compared to women. So we said, oh, that's nicely aligned with our mouse model. And so we then did the three-way interaction of examining sex, injection group over time. And we saw very focal effects, but nonetheless, we see sort of strong effects for the male mice that received the human fibrils. So again, looking at a peak voxel in our injection site, the right striatum, we see the steepest rate of decline for the male mice that received the human fibrils. So we then said, well, given that we're looking at this prion-like spreading hypothesis, how can we examine groups of voxels in which are atrophying together to try and get at the spreading across the brain's connectome? So that's where we use this technique known as non-negative matrix factorization, which essentially allows, as I said, to examine clusters of voxels in which share a covariance pattern. And so the way in which this works is we have an input matrix, which is composed of the volumes at every voxel for every subject. And this input matrix gets decomposed into two matrices. The first matrix describes the clusters of voxels in which share a covariance, and then we can then plot those voxel weights onto the mouse average brain to examine the spatial patterning. And then the second output matrix describes how each subject then loads onto each of these patterns. And we can use linear models to examine whether there's group differences in terms of the subject weight, such that we can examine patterns that have higher specificity to the mice that receive the fibrils compared to the saline injected mice. So we performed this analysis at the 90-day time point. So as I said, once motor symptom onset is nearing, but hasn't quite hit all of the mice. And so we ran NMF with 6 components. So here I'm showing all 6, but I'm only going to focus on component 1, given that this is the only component where we saw this group difference in terms of the subject weights, such that this patterning had higher specificity to the mice that received the fibrils. And so now this color map is denoting the strength of the covariance for every voxel with the full pattern. And so as you see, again, encapsulating our injection site, basal ganglia areas, frontal cortices, and as well as posterior midbrain areas. But when we took a look at whether there's any sex differences here, we didn't see a significant effect for the interaction of group and sex in terms of the subject weight. But we did see a main effect of sex, so such that the female mice had higher specificity to this pattern compared to the male mice. So then we said, okay, how can we look at sex differences differently at this time point? So what we did was we repeated the analysis, only including the male mice, and then again, only including the female mice, and then assessed whether there is any degree of overlap between each sex-specific spatial pattern and the all-subject pattern that I had showed previously. And the way in which we did that was using the Dice-Kappa metric, which essentially allows us to assess the degree of overlap, where we get a value ranging from 0 to 1, 0 meaning no overlap, and 1 meaning perfect overlap. So when we performed NMF for only the males, again, we obtained only one component where we saw this group difference in terms of the subject weights, component 5, and then similarly for the female-only pattern of component 1. And then when we assessed the degree of overlap between the all-subject pattern and the male-only pattern, we got a degree of overlap of 0.46. However, when we compare the all subject pattern to the female only pattern, now we get a degree of overlap of 0.62. So therefore suggesting that there seems to be some female predominance of the patterns of atrophy at this early perimotor symptom time point. So then we said, okay, well, what's happening at the end of the spectrum once the mice are fully symptomatic? So that's at the 120-day time point. And so again, we ran NMF with 6 components. Now we obtained 2 components where we saw this group difference. So I'm going to highlight those here. But really, I'm going to focus on component 6, given that here we observe this marginal significance of the interaction of sex and injection. So therefore, suggesting that this pattern of atrophy had slightly more significance to the male mice that received the human fibrils compared to any other group. And so essentially, again, the pattern encapsulating our injection site also here some ventricular volume changes that are maybe happening, but now suggesting that at this late symptomatic time point, the atrophy seems to be more male predominant. And so going back to our survival plot, where percent survival across the number of days post-injection, where we only saw the male mice succumbing to their disease, what we saw was at this early 90-day time point, pathology seems to be largely female driven. So essentially, seeming that in the female mice that receive the fibrils, there's this early yet slow disease progression, whereas the male mice, once disease has already hit and the mice are fully symptomatic, now we have this male predominance of atrophy, suggesting that there may have a later disease progression, but a lot quicker and more aggressive. And going back to the human condition, where here I'm showing the incidence rates of synucleinopathies across the age in humans. We see a higher incidence rates in men compared to women. And so we're seeing this nice alignment of our animal model with the human condition. And so I'll end here for time's sake, but a big thank you to, of course, my supervisor, Millar, as well as the members of the CoBrA Lab, the Douglas Animal Care Facility members, our collaborators at the MNI, the Fawn Lab, and the EDU team. And thank you for your attention.